Welcome, it's Rijal Aligado. Today, we'll talk about reverse transcription polychain reaction. But before that, let's take a quick review on the central dogma of life. In our last video, I explained to you the central dogma of life, where in a cell, it has a DNA that is to copy itself with use of DNA polymerase in a process called replication, and that DNA is transcribed to produce messenger RNA by enzymes such as RNA polymerase. And this mRNA is read by ribosomes in a process called translation to produce protein. Today, in our topic RT-PCR, we are going to take the messenger RNA as seen in this flow of information by using reverse transcription that converts it back to DNA. But first, what is reverse transcription? It is the process of making complementary DNA or cDNA from a single-stranded RNA. How cDNA synthesizes from mRNA? So we have here mRNA and it has a poly A tail. This sequence of adenosine nucleotide is needed for priming the reverse transcription. In the first strand synthesis in making the cDNA, we use this oligo-DT primer as it contains a sequence of 12 to 20 thymine nucleotides complementary to the adenosine tail. Primers will hybridize to the 3' poly A tail to the mRNA, then a reverse transcriptase is added. And this enzyme binds to the prime region and initiates the synthesis of the complementary DNA strands. So, the half now contains mRNA and the other half is cDNA. The second synthesis requires substituting the remaining mRNA with DNA, and this happens by using RNase H that introduces next in the RNA strand and provides 3' OH priming site. Now, DNA polymerase 1 from E. coli is used to exchange the nucleotides with deoxynucleotides triphosphate, and during the process, the RNA is partly exchanged with DNA building blocks. Aside from that, DNA ligase also provided from E. coli catalyzes the formation of phosphodiester bonds in the DNA to finally form the double-stranded complementary DNA or cDNA. And that ends our video presentation for today. Thank you for listening.